So let's take a look at this picture that can help those of you who are visual learners to think about this. So here we have the filtrate coming up the ascending loop of Henle, entering the distal convoluted tubule, and then it exits to the collecting duct. So here are sodium ions that are in the ascending loop of Henle. These can be transported out into the peritubular capillaries, and to maintain an electrostatic balance of ions. So when a sodium molecule is going to go in, a potassium or hydrogen ion are going to come out. So basically, there's an exchange to keep the charge equal, the net electrostatic charge equal. So you're bringing in a positively charged molecule, so you got to get rid of a positively charged molecule. So it seems that potassium and hydrogen are competitors. So it's either one or the other that's going to be transported back into the distal convoluted tubule. So sodium and potassium are I think of them as competitors for leaving, and that kind of explains why the levels of hydrogen ions can affect the level of potassium ions and vice versa for acidosis or hyperkalemia. In terms of hydrogen ions that can cause the acidification of urine, I'd like to show you an image kind of talking about that. And at first, I want to go back to the proximal convoluted tubule because bicarbonate is picked up from filtrate in the proximal convoluted tubule. And I'd like to talk to you about how that happens. So here we have bicarbonate in the proximal convoluted tubule. This is coming from the Bowman's capsule post-filtration, and this is in the filtrate. What's going to happen is the bicarbonate is going to be condensed with the hydrogen ion, forming carbonic acid. And then carbonic anhydrase, which is the enzyme that we talked about that's found in red blood cells, is physically attached to the plasma membrane of the cell lining the proximal convoluted tubule. This will then break down carbonic acid to water and carbon dioxide, and then both of these will, water will leave by osmosis, and then carbon dioxide is a gas. It'll just diffuse across the membrane. And then inside the cell, lining the proximal convoluted tubule, carbonic anhydrase will reform carbonic acid, and then the bicarbonate will be carbonic acid will break down to bicarbonate and the hydrogen ion. Bicarbonate will be sent out into the interstitial space to be picked up by capillaries, and then the hydrogen ion can be sent back into the filtrate to then go pick up another bicarbonate molecule, allowing the cycle to repeat. And this hydrogen ion is going to be transported with a counter transporter with sodium. Sodium is going to be transported in, and then the hydrogen ion will be transported out. So moving on to the distal convoluted tubule, right? If an hydrogen ion is going to get down there, it's going to be traveling all the way through the filtrate, through the loop of Henle to the distal convoluted tubule. And we'll have some from there. We also have sodium entering the distal convoluted tubule. We have hydrogen ions exiting. Yeah, so hydrogen can help to form ammonia and also a reduced form of phosphate. So the nephrons cannot produce urine with a pH below 4.5, and to increase hydrogen ion secretion, urine has to be buffered. So the buffers I just showed you were the phosphates and the ammonia. They help to buffer urine, and this is important because we don't want the cells in the carrying the urine out of the body or the bladder to be damaged by the low pH. So the way to get rid of the hydrogen ions is to carry them on reduced phosphates or ammonia, as shown here. And these two molecules critically allow the safe passage of the hydrogen ions in a situation where the hydrogen ions are not going to be causing damage by having too low of a pH as the urine is traveling through the bladder to exit through the urethra. So the last topic of this lecture are hormones that affect the kidney. So what I want to look at now is just this diagram of the nephron, right? We have the Bowman's capsule, the proximal convoluted tubule, the loop of Henle, right? And then we talked about the distal convoluted tubule. This filtrate is going to enter into the collecting duct, and the collecting duct, in the collecting duct, there's an opportunity for the body to pick up more water out of the collecting duct. And it uses this by having aquaporins in the collecting duct that are regulated by a hormone called ADH. So if ADH is around, the aquaporins will be abundant in the membrane of the cells lining the collecting duct, and then water will exit. So now I want to show you the mechanism about how this would function, because these aquaporins can be expressed or not, or they can be present in the membrane or not present in the membrane, depending on the presence of ADH. So what does ADH do to these aquaporins? So the cells lining the collecting duct have vesicles containing the aquaporin proteins that are shown. These are transmembrane proteins shown in purple. They form channels allowing water to exit these cells, exit or enter the cells. Okay, so depending what size of the side of the membrane they're on, either the apical or basal side. But in any case, this is a vesicle. What ADH does is it facilitates the fusion of the vesicle with the membrane of the cell. And what we have when ADH is around, we have the plasma membrane containing aquaporin channels. So let's say there's no need for ADH to be produced, then the antidiuretic hormone will be not expressed. What's going to happen is the aquaporins that are near each other in the plasma membrane are going to perform a process very similar to, they're going to perform endocytosis. Endocytosis will be performed, bringing in these aquaporins, and it'll restore to the vesicle. So what we're seeing now is basically the process of endocytosis to bring these transmembrane proteins back into the vesicle, and we're seeing a process like exocytosis to bring this vesicle to the plasma membrane, except that the contents are not going to be released like we usually have in exocytosis. Their final destination is just being in the plasma membrane. So let's take a look at the this in a diagram. So if you have a situation where you're not drinking enough water, you will become dehydrated. Dehydrated means there's not going to be enough water, and just like you had seen in the descending loop of Henle, as water leaves and the solutes stay, you're going to have an increased plasma osmolality. Then we have osmoreceptors in the central nervous system. These osmoreceptors in the central nervous system will send action potentials down to the pituitary gland, and that will then be a signal to secrete the hormone, antidiuretic hormone. Okay, antidiuretic hormone will be produced. That will then go to the kidneys, and when ADH is produced, it is going to lead to the aquaporins being sent from the vesicle into the plasma membrane. This will allow increased water reabsorption in the collecting ducts. Therefore, less volume of urine will be secreted, and the urine will be more dark yellow because there's more solutes the solutes are more concentrated in a urine of someone who's dehydrated. So if someone always is urinating with the yellow color, that means they should drink more water. The opposite situation happens when you're drinking a lot of water. Okay, so you're overhydrated or well hydrated. If you drink a lot of water, you're going to have a reduced plasma osmolality. The osmoreceptors will detect that. It'll send signal to the pituitary gland to stop producing the antidiuretic hormone, and basically that will lead to the aquaporins to be endocytosed into a vesicle back into the cytoplasm of these cells lining the collecting ducts, and then you'll be urinating in bigger volumes, and because the solute is going to be more dilute, thanks to having more water in urine, the urine will be either clear or very, very faint yellow. And I should also mention that another name for the antidiuretic hormone is vasopressin, and it got that name because it helps to increase blood pressure. And then last but not least, I'd just like to show you a table that helps to reinforce some of the concepts I've just have discussed. So as we had mentioned, under dehydration conditions, you'll have higher osmolality in the blood. Osmoreceptors in the brain will detect that. They will trigger the pituitary gland to increase the secretion of antidiuretic hormone. This is going to send a signal to the cells in the lining the collecting ducts that they need to send the aquaporins and vesicles to the membrane to allow water to be leaving the collecting duct and to enter the interstitial tissue. That will lead to a reduced urine volume. In contrast, if we have reduced osmolality in the blood, the osmoreceptors will detect that, and it will then reduce and inhibit the secretion of antidiuretic hormone. We know what that's going to do is it's going to basically signal the aquaporins in the plasma membrane to endocytose, back to being a vesicle. That means water is going to stay in that collecting duct and not travel into the interstitial fluid or interstitial tissue. And then the amount of urine, the volume will be increased. So let's now look at blood volume. Okay. So if you have an increased blood volume, right, but the solutes are the same, you're going to have a reduced osmolality. I just want to make that connection for you. So reduced blood volume would be also having the increased urine because you're having decreased antidiuretic hormone. And then as you urinate more, your blood volume can be decreased. In contrast, you have the reduced blood volume, right? When you have reduced blood volume, that means you're going to have more solutes because the solutes are the same just in a smaller volume of liquid, and that's going to be then the situation of higher osmolality, right? You're going to have increased secretion of ADH, meaning those aquaporins are going to be expressed in the collecting ducts, and then the water is going to be leaving into the interstitial tissue, and then we're going to have a decreased urine volume. So thank you for your attention, and I hope this is interesting to you as it is to me, and that hopefully this all comes together with thinking about this. I think this is a good table to kind of test your self on understanding the role of antidiuretic hormone and how that affects the volume of urine. Take care.